Hello, everybody. I'm Charlie Doring, the director of the Center for the Study of Complex Systems here at the University of Michigan, in case you're coming from outside the University of Michigan. And today, we're holding our Nobel Prize Symposium. This is an event that we've been doing in complex systems for a number of years, almost continuously for probably most of this century, to tell the truth. I think we missed a few years, depending upon details. And the point of this event is to utilize the expertise at the University of Michigan amongst the faculty and research staff to try to explain to the rest of us what this year's Nobel Prize in the various categories was about, who won, why this is interesting for their field, and maybe, you know, in many cases, there are personal connections between the faculty here at Michigan and the prize winners, and learning a little something about the prize winners themselves. And so this is it's a fun event, and what we're going to do is there will be time for questions and discussion, which is a lot of the fun of this. And we will take questions, and Linda will help me with this. We will take questions in the chat, or
people can put questions in the chat, and then at the end, either if you want to call on them or if they really want to speak themselves, put their hand up and we'll unmute you.
Right. We'll do that, and we'll do that as time allows. Right, because we have a pretty tight schedule here, and yeah, so that's that. And I'm looking at the clock, and we're just about right to introduce the first speaker. And so in this case, we will. Excellent. We've got the first slide coming up here. Now, the first subject today is the Nobel Prize in Chemistry. Which might sound a whole lot like biology to some people. This is also an interesting thing about the Nobel Prizes. And our presenter is Nils Walter, who is the Francis S. Collins Collegiate Professor of Chemistry, Biophysics, and Biological Chemistry. So he has joint appointments in LSA, Literature, Science, and the Arts, in the medical school. And I think the time, well, let's see, I got to talk for another 30 seconds or something, according to this. Anyway, Nils will set the pace going here. And Nils, I will give you, I will definitely interrupt after 25 minutes. And if you end earlier, that's fine. We'll just start with the questions at that point. Okay, my clock says 1:15. Nils.
Excellent. Thank you very much, Charlie, and the Complex Center for the invitation. It's a particular pleasure and an honor, actually, for me to talk about what I think of the CRISPR craze. And I want to use this talk today to tell you how scientific breakthroughs come oftentimes to the prepared, but when least expected. And it's a particular pleasure for me to give this starting talk of the symposium, because Jennifer Doudna is a good friend of mine from the field, and now her best friend, Emmanuelle Charpentier, of course, the two of them together got the Nobel Prize for Chemistry this year for the development of a method for genome editing, as indicated by the Royal Swedish Academy of Sciences. And as you can see, Alfred Nobel looks very favorably at these two women. And much has been made, actually, of the fact that while this chemistry Nobel Prize has been given for 120 years now, 1901 it started, has not ceased even during the two wars. And roughly 200 people shared them, initially just single people. More recently, it's oftentimes 2 or 3 at a time that get it each year. Among the 200 or so awardees, only 7 have been women so far. And also what has been noted by the committee and many others, that it's the first time that 2 women share this prize. And so that's a particular breakthrough in itself. But I want to focus instead today on the question of what leads to these discoveries. But first, I want to tell you a little story that Jennifer herself tells you. Here is a little snippet from, or a little snapshot from an interview she gave to NIH Director Dr. Francis Collins, just a day or two after the Nobel Prize was announced to be given to her and Emmanuel. And I mean, I won't play this for you. I encourage you to hear it. It's a great interview. But Jennifer, as you can tell, told a couple of funny stories. One of them is how she actually learned about the Nobel Prize. As you can imagine, there's a 9-hour difference between Sweden and California at UC Berkeley, where Jennifer is located. And the Swedish Academy actually did not reach her early in the morning or late at night, if you wish. And instead, the first person to give her a call was a journalist. And the journalist says, "So, what do you think of the Nobel Prize?" And she said, "Oh, it has been given. That's great. Who got it?" Only to then realize that it was actually her, and she had to break off that phone interview with the journalist and get on the call with Sweden to get it confirmed and figure out what has happened. So sometimes people are unaware or not prepared for what happens to them in good news. Hopefully we'll have good news for us as well. RNA might be the vaccine solution for the current pandemic, of course. And what I wanted to tell you then is a little bit of a background of the science that Jennifer and Emmanuel discovered. And to do that, I will actually share a different presentation here, a little movie that I found that was made by one of the institutes that was involved heavily in discovery, actually at the MIT. And so I'll share a quick movie on that from the internet here with sound that runs about 4 minutes and just explains the science to you in quite nice ways, I think. So you should be able to see that now.
Every cell in our body contains a copy of our genome, over 20,000 genes, 3 billion letters of DNA. DNA consists of 2 strands twisted into a double helix and held together by a simple pairing rule. A pairs with T and G pairs with C. Our genes shape who we are as individuals and as a species. Genes also have profound effects on health, and thanks to advances in DNA sequencing, researchers have identified thousands of genes that affect our risk of disease. To understand how genes work, researchers need ways to control them. Changing genes in living cells is not easy, but recently a new method has been developed that promises to dramatically improve our ability to edit the DNA of any species, including humans. The CRISPR method is based on a natural system used by bacteria to protect themselves from infection by viruses. When the bacterium detects the presence of virus DNA, it produces 2 types of short RNA, one of which contains a sequence that matches that of the invading virus. These 2 RNAs form a complex with a protein called Cas9. Cas9 is a nuclease, a type of enzyme that can cut DNA. When the matching sequence, known as a guide RNA, finds its target within the viral genome, the Cas9 cuts the target DNA, disabling the virus. Over the past few years, researchers studying this system realized that it could be engineered to cut not just viral DNA, but any DNA sequence at a precisely chosen location by changing the guide RNA to match the target. And this can be done not just in a test tube, but also within the nucleus of a living cell. Once inside the nucleus, the resulting complex will lock onto a short sequence known as the PAM. The Cas9 will unzip the DNA and match it to its target RNA. If the match is complete, the Cas9 will use 2 tiny molecular scissors to cut the DNA. When this happens, the cell tries to repair the cut. But the repair process is error-prone, leading to mutations that can disable the gene, allowing researchers to understand its function. These mutations are random, but sometimes researchers need to be more precise, for example, by replacing a mutant gene with a healthy copy. This can be done by adding another piece of DNA that carries the desired sequence. Once the CRISPR system has made a cut, this DNA template can pair up with the cut ends, recombining and replacing the original sequence with the new version. All this can be done in cultured cells, including stem cells that can give rise to many different cell types. It can also be done in a fertilized egg, allowing the creation of transgenic animals with targeted mutations. And unlike previous methods, CRISPR can be used to target many genes at once, a big advantage for studying complex human diseases that are caused not by a single mutation, but by many genes acting together. These methods are being improved rapidly and will have many applications in basic research, in drug development, in agriculture, and perhaps eventually for treating human patients with genetic disease.
All right, so this, of course, is the science behind the discovery, but I did want to spend more time on actually the background of what led to this discovery and how it actually can be used in various different ways. And this to me is fascinating because it shows how science builds sequentially with discovery, after discovery after discovery. And we all stand on the shoulders of giants in order to do the science that we do today. If you are interested in watching more movies, here's another one that is actually a presentation by Jennifer Doudna herself, kind of technical, but in detail to the Swedish Academy of what the discovery actually entails. Where I want to pick up is, again, the history and how strong foundations and patience lead to breakthroughs like this one. And I want to focus you first on the idea that there was a CRISPR biology only since 1987, so a little over 30 years ago, where CRISPRs were first described. They actually had a different name at the time. And then over a period of a few decades, couple decades, additional discoveries were made until it was realized that RNA and an enzyme called Cas9 plays an important role in these repeat sequences that are called CRISPR. And that then led to a seminal paper in 2012, so 8 years ago. It only took the Nobel laureate committee 8 years to figure out that this was important, because Jennifer and Emmanuel actually discovered that you can combine this CRISPR biology arm here in cyan with a genome editing arm that had been independently ongoing, totally unrelated to the CRISPR biology for many, many years. Where starting in 1979, so something like 40 years ago, the first time a gene could be replaced in yeast, and then various different proteins were engineered over time in order to make that more precise. But it was a very, very clunky technique until CRISPR-Cas9 came to the fore. And after suggesting that genome editing would be possible in 2013, then a number of groups actually showed that this can be done. And from there on, many, many more papers ensued. But I want to take you first to 1987 and to Japan in particular. In the 1980s, of course, lots of technology came out of Japan, from the Walkman to Nintendo video games and all that. And so it turns out that basic science is sometimes done in various places. And basic, in a sense, is actually the wrong word, because it's foundational science that really it represents in hindsight. And this goes back to this paper from 1987, in the Journal of Bacteriology, so a specialist journal on bacteria, that in this, by Nakata, this Japanese group, they discovered that there is an unusual structure in the 3' end, so on one end of a gene, flanking this gene IAP or YAP, where they find highly homologous sequences of 29 nucleotides that are arranged as direct repeats with spacing in between. And the repeats are shown here. You can see, if you look from top to bottom, that they're the same sequences found in repetition in the same genome of the same bacterium here. And there was no homology between the spacings in between these repeats. And so therefore they concluded that because there's no homology found so far between this particular bacterium and others in regard to these spacers, the biological significance of these sequences was not known. Okay? Fast forward to 2000, a Spanish group under Francisco Mojica had worked diligently to find other such repeat sequences in a number of different bacteria, and they wrote a little micro-correspondence, so just a letter to the editor, essentially, in this, again, specialist journal, Molecular Microbiology, that said that a peculiar type of repeated element has been detected in different prokaryotes and different bacteria, and they seem to have similarities between different phylogenetic groups, which is different bacteria that are not evolutionarily related and are far distant from one another. And you can see an alignment of these different bacteria and the particular sequence in them here, these repeat sequences. You can see that everything that's underlaid in black is similar between the different genomes that now had been sequenced. And Mojica went forward and called these short, regularly spaced repeats, or SRSR, not particularly well pronounceable, as you can imagine. Another 2 years later, a CRISPR name was invented, so a better name was invented, and this goes back to a group in the Netherlands, a Dutch group under Ruud Janssen here at Utrecht, who found that not only are these repeats found between different bacteria or prokaryotes, but they're actually associated with particular genes. And these, what they called 4 CRISPR-associated or Cas genes, were identified in CRISPR-containing prokaryotes and absent in CRISPR-negative prokaryotes. So sequences, bacteria that don't have these repeats would not have them, but those that have the CRISPR repeats would have these CRISPR-associated genes, suggesting that there's a biological link and opening up the possibility to discover the biology of these repeat sequences. And this is illustrated from a figure from this paper here. So you see the repeats here in the black bars. This is part of the genome sequence of this particular bacterium. Here I list just 4, but there are many others in this paper. And you can see these arrows indicate different genes read out from left to right or from right to left in some cases that are these Cas or CRISPR-associated genes, and that are always found flanking the CRISPR repeats here in the middle. Right? And so that, even though it's not exactly the same genome structure, is similar between these bacteria. The next similar discovery came in 2005, where it was found that the CRISPR sequences originate actually from past viral infections. No wonder that the sequences, the spacer sequences in particular, are not found between bacteria, different bacteria, but oftentimes only found between bacteria to be similar to those in phages or viruses of bacteria, and that they provide bacterial cell immunity via RNA, ribonucleic acid, the molecule that was described in the video. And this discovery was made in France in particular by the group of Duska Ehrlich. And he described that these spacer elements are the traces of past invasions of these extra chromosomal elements, that is phages that have infected the virus, the bacterium, as you saw in the movie. And they hypothesized that an antisense RNA, ribonucleic acid, is read out from that spacer sequence from the genome to make the bacterium essentially immune against future or the next infection by this particular phage. And the key piece of data is shown here, looking at the proportion of propagating phages or viruses, independence of the number of spacers. So if you have lots of repeats of these spacer sequences that are all aligned with the phage genome, with the viral genome, if you have a lot of them, then you get very little infection with this virus, indicating that it's really mediating immunity, kind of an intracellular immune response of the bacterium against the virus, against the phage. And I should note that at this point, not only a French group, but a second French group at Mojica, Spain, all found very similar things. So steam is picking up, things are getting faster, more groups are getting involved at this point in the middle 2000s. In 2008.
Here's your time warning, Nils.
Okay.
Okay.
It's 16 minutes by me? Okay.
Okay. 10 more minutes.
Yes, 10 more minutes. Okay, fine. Yep, that's exactly right. And so in 2008, it was discovered and published by a group in Netherlands in a much more high-profile journal in Science by Jan van der Oost, that CRISPR RNA guides are actually what mediate the antiviral defense in prokaryotes. And there's a key piece of data here again, where if you have more of these CRISPR repeats and make more of the RNA, then the sensitivity to a phage goes down. The bars become shorter here in this depiction. And around the same time, also published in the high-profile journal Science out of the US, Eric Sontheimer, who was actually at the time at the University of Chicago, found that CRISPR, what he called then CRISPR interference, limits not only phages, but also horizontal gene transfer. So what bacteria can do is if one bacterium knows how to fight against an antibiotic, then it can share that genetic information with the recipient cell through a little tube where the DNA is pushed through. And this conjugation can lead to distribution of genetic information. But if the recipient cell, the bacterium that gets the DNA, is not interested in it, it can actually make CRISPR repeats against that DNA so that in the future it doesn't get infected or transferred such in this horizontal gene transfer such information. So it can make a decision to defend itself against another bacterium, giving it genetic information. And so Eric showed this, and so this was important because it showed that there is really a broad relevance of these genetic elements. And then for the first time, Emmanuelle Charpentier came to the fore here. At the time, she was associated with Sweden and Austria in particular. And she showed that CRISPR RNA maturation is done by a trans-encoded small RNA. And this is shown here. She called that a trans CRISPR RNA, which is short a tracer RNA here, ribonucleic acid. And this RNA binds to the precursor of the CRISPR RNA that has been transcribed from the CRISPR repeats. And that then binds a protein and gets processed. And what remains is a much shorter guide RNA that has this sequence in green that's from the original phage or virus infection, and then in black, a repeat sequence that is used to shuttle it between the genome and the RNA versions of it. And I should say that another friend of mine, Jörg Vogel in Germany, was actually also involved in doing some of the transcription analysis that was necessary. Now comes 2012, where CRISPR-Cas9 actually was discovered to be able to use a single guide RNA. And this is really where Jennifer apparently got a call from Emmanuelle. Emmanuelle Charpentier was in the Bay Area near San Francisco and near Berkeley, and gave her a call and said, look, you're an expert for RNA. Can we work together on this tracer RNA that I just discovered? And they realized that they actually can make a construct, a protein here in blue that binds the DNA. So the protein is this Cas9 version of the CRISPR-associated gene. And then it binds the target by way of this CRISPR RNA and the tracer RNA that I showed you on the previous slide. But they realized that you actually can combine the two by just connecting them, these two ends here, with one another through a linker loop, as you see here. And then all of a sudden, that there's a possibility of using a single RNA-guided Cas9 to potentially do a programmed DNA cleavage and genome editing. So cut the genome where it's needed. And that actually was possible and was demonstrated to work in cells, in human cells in particular, by 3 groups now, all in the US, including Jennifer Doudna's group, who published technically first on this on January 29th of 2013, that showed that you can use this RNA programmed genome editing in human cells. And then in competition with her, 2 groups from Cambridge, Feng Zhang at MIT, as well as George Chert at Harvard, showed something very similar. And you can tell that, I mean, this came 2 weeks before the pair of Science papers here on February 15th. And so that shows you how close that race became. And it was all in high-profile journals, indicating that people caught on to the importance of this. Now, what's the lesson from this history lesson? Well, going from curiosity-driven foundational research, like finding these strange clustered regularly interspaced short palindromic repeats, giving them a good name, the CRISPR name, in bacteria, to then finding a way to edit the genome, right, took only 26 years, or it took a quarter century, over a quarter century. So it takes some patience, it takes some time, and you never know where a similar discovery in the past might really lead to the next breakthrough in medicine and chemistry. And you could ask, is that it? No, I would say the revolution is still ongoing. This is from a review just a couple of years after the seminal discovery by Doudna and Charpentier, where they outlined that you can manipulate the Cas9 enzyme here. You can make it to cleave only one of the two strands of DNA instead of both of them. And then you can do something where you put two of these Cas9s with different guide RNAs together to cleave a longer piece of DNA out of the genome, as you see here, cleave one strand here and the other strand here, and then the whole thing can be cut out or repaired. You can also pull other effector domains, other proteins to a DNA based on the guide of the Cas9 system, the CRISPR-Cas9 system. And I should say that we owe a lot in this respect to bacteria. Bacteria have not just this one type of Cas9 enzyme, as it's found in class 2, type 2, but also many, many other versions that have found other applications. Some of them use shorter, some of you use longer sequences, shorter sequences to recognize in the genome, makes it easier to find or to versatilely find different sequences that you want to cut. A longer one gives you higher specificity. Both can be important in certain circumstances. And this is just a chart that shows just how many of these different classes are shown, where the size of the circle here indicates how many are already sequenced from the different bacterial genomes we have. And then the future of CRISPR-Cas9 is definitely very bright, potentially in human gene therapy, screen for drugs, and agriculture, and many other things potentially. And here are just some examples, and I won't go into the details, but in addition to editing the genome, you can also light it up, for example, by putting a little light onto your CRISPR RNA or your Cas9 protein, and then localize that sequence in the genome, in the whole, in a cell, in the nucleus of a cell where the genome would be. And so you can do fancy things to modify and mark different versions, different parts of the genome. And that has led to this explosion in number of publications in this field, and it's only ongoing. And of course, the Nobel Prize will only accelerate this. Now, I also wanted to tell two side notes. One is that there's always a flip side. There is actually a long-running patent fight between the MIT group and then the Berkeley, Vienna groups of Charpentier and Doudna. And in principle, the problem is that one person invented the wheel, but the other person invented the patent, right? This fight is about whether somebody writing down something in a notebook and then publishing something after the fact is the same or supersedes basically publishing something else first. So, and that is still ongoing here from a recent article, that fight. Of course, there's a lot of money to be made by the universities, potentially Berkeley and MIT in particular. And last but not least, I also want to give you the idea that there's a risky side to genome editing, and ethics need to be involved. And actually, Jennifer Doudna, my personal hero, is actually involved in figuring out how to maneuver this. She herself heads a plant genome editing institute, so on neutral ground, if you wish, no humans involved, but also is a pioneer in thinking about how to ethically utilize CRISPR-Cas9 for genome editing, what you can and cannot do. So in summary, I want to drive home the point that basic science is oftentimes serendipitous and foundational, and needs and deserves long-term support, because patience is rewarded in the end after 26 years in this case. It takes a village to raise a child, as you've seen with my little excursions across the world, from Japan to Spain to Holland to the US. Lots of different places were involved in finding all the different things that were needed for this discovery, and we all stand on shoulders of giants that came before us. And then, of course, great opportunities come with risks that need to be mitigated. We are at a place as biologists now where physicists were something like 100 years ago, when they understood that they can split the atom and had to decide what to do with that. Here we can split the genome and need to decide how we properly handle this in an ethically correct way and help everyone on Earth with this. And so that's all I had for you, and thank you very much for your attention.
I'm going to applaud. Thank you, Nils. That was fantastic. And I apologize for jumping in 10 minutes early. You know,
yeah,
I'm getting warmed up on this business.
No problem.
So everybody in the audience, you know, you have your little reaction button down there. And so we'll be counting, Linda will be counting how many applause hands everybody gets. No, we'll keep score. So we have time for questions, and I don't see any questions in the chat right now, but I have a question for you. So so these SRSRs, the short repeating sequence, right? This is a fundamental to the process, right? And you said so these require prior viral infections in order to sort of implant these. Now, of course, we are all very sensitive to viral infections these days, but I'm going to guess that there are all sorts of very sort of benign viral infections that happen all the time, sort of generic viral infections. Could you say something about that sort of prepare all of our cells to be susceptible to this procedure?
Yeah, so for one, clearly these repeat sequences with the spaces in between that are then from the phages are fascinating in that means that a previous ancestor of that bacterium got infected by that virus. And not all the bacteria got killed by it. At least one of them survived, and that then basically used that sequence, put it into its own genome so that its progeny, the following generations, would no longer be infected by this virus. So it's a kind of a near-death event. The atom bomb went off, the phage amplified and almost overwhelmed the bacterial cell, but the bacterial cell learned from it and passed that on to future generations. These generations, these sequences are kept for hundreds of generations of bacteria and eventually are lost if no new infection occurs, but clearly are important to prepare for the future. And in fact, something similar, of course, has happened with our genome. There's evidence that up to 8% of the human genome is actually retroviral in nature. That is derived from viruses like HIV and other retroviruses, which basically have a genome that's RNA and that they can make back into DNA, as we call reverse transcribed into DNA, and then put it into our genome. They disrupt genes, but also enrich our genome, as it turns out. And so it's not entirely clear why, but we do accumulate viral sequences also in our genome. Whether they actually protect in the same way is certainly not with the CRISPR-Cas9 system, but they may be involved in regulating gene expression and make us more adaptable to changing environments. So not all viral infections are bad. Not all are as bad as the current coronavirus. It's very unique in terms of how quickly it affects and how low ultimately a death rate it has, because Ebola has such a high death rate that it doesn't go very far in terms of infections. But this is kind of a happy or a bad medium for the virus in that it replicates well, but is sufficiently benign and keeps people happy for 2 or 3 weeks before they come down with it, that they can affect a lot of people. So as far as viruses go, the coronavirus that we have now, SARS-CoV-2, is probably the worst it could get. But it's likely that we'll have more viral infections. But again, I would bring this back to the idea that not all viral infections are bad. There can be actually positive aspects to it. And ultimately, our genome exchanges information, genetic information with the environment that includes viruses over time. Right. So that's how we evolve.
Right. Yeah. Thank you. Okay. We have a couple questions in the chat. Okay. So here's one. This is from Matthew Culberson. For those uneducated people who say, quote, "This is biology, comma, not chemistry," unquote, how do you respond to them?
Yeah. So the chemistry Nobel Prizes have vacillated between synthetic chemistry, so classical chemistry, you make a molecule, you make and break bonds between carbon atoms and make a new molecule, and more applied biochemical prizes. And I mean, it goes back and forth. There's definitely the committee of the Royal Swedish Academy certainly has a lot of discussion of where it should go each year. And there's a big competition for it. Actually, the Nobel laureates have to give presentations in Sweden. I mean, the future Nobel laureates, and they all see each other and they compete with one another. It's kind of a Shark Tank type of experience, I hear. And so then each year a decision is made, but it has, I mean, oscillated between these two extremes, if you wish. My argument, of course, is I'm a chemist in the chemistry department, but I'm actually a biophysical chemist, as you alluded to earlier. And I think that's the future of the basic sciences, of the natural sciences, to grow together those different areas. And I think it's important for us to understand that fundamental things that are chemistry happen with these molecular scissors that we now have, right? With CRISPR-Cas9, hydrogen bonds are made, base pairs are made. That's fundamentally chemistry, and that's the basis for it. So for me, chemistry, and for other people, chemistry is the central science. So I think it's good for us as chemists to actually appreciate the flanking areas of biology and physics and include them in our considerations.
Right, I agree. I think we have time for one more question here. Lan Xing asks, can we use CRISPR to edit massive amounts of cells at the same time? And if so, are there risks if the editing fails?
Yeah, so this is done in the test tube now, where you make thousands of different guide RNAs. You need only one guide RNA to target a particular piece of the genome. There are tricks with which you can actually edit in a large number of cells, each cell with its own guide RNA only, with a single specific guide RNA that's different from the cells in the pool that are edited by another guide RNA. So there are tricks that people use along those lines. So yes, it's technically possible. And sure, so one way to think about this is going back to DNA technology, right? So once we can take apart DNA, what happens with it? Can that be a good or a bad thing? On the other hand, nature does it all the time, right? As I said, there's horizontal gene transfer. Phages or viruses infect us and put sequences in our genome. Our genome sequence picks up sequences from viruses, from the environment all the time. So nature has done this for billions of years. I think it's actually wrong to think that we do anything different than what nature has already done for billions of years. And I think it's actually almost hubris to think that whatever we can do is going to be worse than from what nature has already done. Now, again, you don't want to cross certain barriers. And that's definitely, I mean, where the ethics come in, which is why I emphasize this. But in principle, yeah, I think lots of things have been tried by nature already. So we are fooling ourselves to think this is the first time.
Right, right. But it is a brave new world out there.
It
with this stuff. Yeah. Okay. Thank you very much, Nils. That was great. I'm going to applaud again.
Thank you.
I see Raul applauding in the window.
Thanks, Raul.
And
Thanks,
Doug.
everyone.
Yeah. Okay.
Excellent.